Cytoplasm is not everything inside the cell. It does not include what's inside the nucleus. And so you say cytoplasm is just a light blue area. It's not the nucleus too. So you understand that. Nucleoplasm is a different beast, so to speak. And it is not part of the cytoplasm or cytosol. So, but you saw the slide now that you had to print off, it'll be easier for you. And Hillary thought we might finish this PowerPoint day, and we probably will. That's so short. And I'll just double up Monday and give 2 tests.
Oh, gosh.
Weekends, you got time to do one more. This can't be very big. Oh, I don't know. You know that iodine test, the iodine lab, the test? 15 answers on it.
15 answers?
15 answers on the iodine. And I got it finished last night. There's nothing that I had to grab. There's things that you saw and you should know.
What kind of questions? Multiple choice?
I'll give you pictures of like test tubes and ask you what kind of result that was and things like that.
Okay.
All right. So the next slide, is this a new slide for y'all?
Yes.
Okay, so I'll give you time to copy it. I didn't think you saw this one yesterday. I see one thing I want to fix now. I need to put a space. Those are too close together right there. No big deal. It bothers me.
Cytoplasmic needs to be read.
Oh.
Cytoplasmic before filaments.
Oh, that's red also? Okay. Very good. Oh. I want to talk about this a little bit. That cytoskeleton up there does a lot like what your skeleton does for you. Because yours is made of bone, that's made of protein. The cytoskeleton, it can't be bone, of course, because bones are made of cells, and these things are inside the cell. So it's made of protein. And you know, we mentioned, you do know sperm has a flagellum, right? It makes it swim. Well, that flagellum is an extension of the cytoskeleton, and those pigments do this over here, and that makes it whip back and forth, and it swims. The shape of the cell, whether it's round or squarish or star-shaped, is also because of the cytoskeleton. Even our shape, you know, we have a basic human shape, and the dog has its basic shape, and the cat has their basic shape, and the horse. It's all because of bones. It's all because of skeleton. The skeleton. So in this case, the cytoskeleton is similar in function for a cell as your bones are for you. That's why it's called cytoskeleton. There's no bone in that cytoskeleton. It's made of protein. Also in that cytoplasm, you'll find nutrients that have been dissolved. It also, it also gets rid of waste products. The cell makes trash just because it is living. It needs the goodie to keep living, and then using the good. Well, even your house produces trash. When you cook a dinner, you got boxes throw away and wrappings throw away. So even though you're preparing dinner, you're going to produce trash products. The cell's the same way. It's got trash. It's got to get rid of it. But then again, if you stop going to the grocery store, that's bad. If the trash man has a strike and don't come for a couple of months, that'd be terrible. Okay? So anyway, the cytoskeleton is, on this slide here, the cytoskeleton is the featured event. And it's the bones of a cell. No, it's not bones. But it's protein. All right. There are salts dissolved in the cytoplasm. And they're there because a cell must be able to conduct electricity for it to be alive. That's why when you get electrocuted, it is so bad. Your cells are really good conductors. You hit a live wire, especially standing in water, it fries you. And mechanics basically means the way the cell functions, the way it moves, what it does. Cytoplasm is still mainly water, though. Still mainly water, what, 80% you say? It's mainly water. And nothing, when we go and, if we explore the moon to look for life, all we look for is water first. If you don't find water, you're wasting your time because there's no life, because there's no water. As far as we know. There might be some completely different system that Lord made it. It might use alcohol instead of water. As far as we know, based on our experiences, if there's no water available, then life will not exist. And that's just what we know. We don't know what about out there. That's somebody one time said, and they had it real well. Our microcosm, our little world, could be no more than a molecule and a drop of water being looked at by some of the beings. We would be the amoebas. You follow me? It makes you look, that's abstract, but we think about us looking at other things and one was looking at us. We might be the amoebas being studied instead of us studying other things. But as far as we are concerned, in our little world, if there's no water, there will be no chance of life forms to be found. Even ice. If it's ice, that would mean one time it was water. We might find the remnants. I hear on the moon that the part you can see is really, really hot. And on the backside is really, really cold. And right there on the edge where the light hits the dark is the only temperatures that can possibly we could stand. But there's no air on. There's no air to breathe there. So if I give you an air tank, then you're still going to freeze to death or burn up. So when we landed on the moon, we stuck a flag on it. But it's right there on the edge. It's not in the middle. It's too hot there. It's on the edge. Yes.
Is there wind in space?
Is there wind?
Wind gusts.
What you saying?
Is there wind in space?
I don't know.
Because the American flag is definitely blowing in the wind.
Well, I can freeze that, but I can make a flag out of wood and paint it. You see it floating? You see it flopping?
Yeah, like in the old Armstrong, whatever, when he first got on the moon, the flag is definitely going like this.
I don't know that.
But there's no air in
Because they
space.
did it
Yeah,
at the
yeah.
sound stage.
If there's no air, how can there be wind?
Exactly, that's
It's
So,
a
why.
I don't
soundstage
know.
in It's that conspiracy.
I don't recall actually studying the picture of Armstrong. I always thought the flag was like steel,
It's
like a piece of starch cloth that would not float around. Yes.
Aren't there storms like on Jupiter and stuff? How does that happen in there?
Well, I don't know. I'm not an astronomer. You asking me? I have no clue. I know there are storms and raging. And can there be wind without oxygen?
Yes.
I mean, it matters what's being blown.
Technically, do we need the air or the oxygen? We need oxygen. I guess it could be windy up there, but the wind contains no oxygen. I'm still in trouble. And I know what you said. I know that's true. But now I think Saturn has like a big hurricane going on all the time.
Don't they have their own atmospheres, don't
They do. They do. And anything that we living there must be adapted for that environment. We couldn't live there unless we modified our spaceships. We live in a spaceship. Actually, when you go into space, we carry a dab of Earth with us, don't we? Our environment, our air. If we give out of air, then we'll die. If we give out of food, we'll die. I understand that when they come back from space travel, they have to be brought out in wheelchairs because they're so weakened from no gravity, the muscles have lost their strength, and they actually can't walk down the ramp, they fall. So they rolled out in wheelchairs. I've seen it happen one time before. And it didn't dawn me at the time why they're in wheelchairs. I guess because then we thought about having a colony on the moon where people would live there and have children. But my thought was, well, they never come back to Earth and visit grandparents. The child born in what and when they grow up to be as big as I am, genetically, yes. So could their muscles handle an environment where they're 6 times heavier than they really are? On the moon, you're 6 times lighter. That's why you can walk around so weird looking. And you come back, if you're born up there though, and you're used to being in that, when you got back here, you'd be 6 times heavier. A 100-pound person down here would weigh 600 pounds. Could they even stand up? How about the heart, the strain on the heart? I would not want to be in that first number that went to the moon for any amount of money. To be a colony. They thought about, fresh air, but you had to grow your own plants, of course, because plants will make the air for you. But those are all good philosophies and things to think about. I see up there eukaryotic cell. Do you know the other kind?
Prokaryotic.
So prokaryotic is the other kind. Between the U and the pro, who's the oldest?
The pro.
The pro is the oldest, like a prototype. Eukaryotes came on the world later. Prokaryotes, and we still got prokaryotes. But if you go back long enough, if you went back in time, you go back to a place that there's only prokaryotes, because eukaryotes have not come on board yet. The claim to fame of eukaryote, all this stuff is in a membrane. It's got membranes around the mitochondria. It's got membranes around the lysosomes. It's got membranes here. Got prokaryotes have no membranes other than the cell membrane. But inside there, you don't see a nucleus in a prokaryote. There's no nucleus. It's just blah. All the stuff is there. All the stuff is there. DNA is there. It's just floating everywhere. It says protoplasm. We're trying to change that word to protoplasm. We're trying to say cytoplasm. And some folks say that when you say protoplasm, that means everything, including nucleoplasm. If I say cytoplasm, I'm eliminating the nucleus and all the content. When I say protoplasm, I mean everything in that cell. Everything in that cell. It says the cytoplasm and the nucleus, those two together, well, that's that's everything. So, if I ask you to do like a dichotomous key, you would have protoplasm, and a line going down says, and some is cytoplasm, and some is nucleoplasm. But all together, it's protoplasm. And all cells have protoplasm. Now here's the other kind that I'm glad you knew about. Their nucleus is not, it don't just leap out at you. They do have a cell membrane. Because you got, that's how you maintain a cell. But other than that, there's nothing inside that you don't see any structures. All the stuff is there. It's just not packaged in the packages like a eukaryote. This is very primitive. Now, as you become more organized, you become more advanced. This cell is so disorganized compared to eukaryote where everything's in little bags. This thing right here, there's no bags inside there. All you have is cell membrane. That keeps the cell together. That's it, baby. That's it. So the cytosol would contain all the stuff that cytoplasm has, plus in this cell, the chromosomes are there, the nucleolus is there. I mean, they're not contained in a thing called the nucleus. They're all just floating around there together. I'd much rather study the eukaryote. There's more stuff to see than the prokaryote. There's a region called the nucleoid. It's like a blemish. It's not really maintained by membrane, but you can say that there's something right here. You say a lot of dark stuff right there, and there's no membrane surrounding it. You can't tell where it bleeds away, but that's the nucleoid region. And you'll find chromosomes there. Now, the next, when I do go to my next lesson, it's about the organelles that's in the cytoplasm. But this lesson here is only about the cytoplasm. I'll tell you right now, though, that the organelles, the mitochondria, the reticulum, the lysosomes, the peroxisomes, are all floating around inside the cytoplasm. There are two micro things, microfilaments. That's a solid chunk of protein. And a microtubule is like a round straw looking. It's hollow. Now, they're both micro means your eyeball can't see it. You can't even see a cell, let alone the stuff inside a cell. We're talking about things seen by the electron microscopes upstairs. Can't see this stuff. That has been examined with the electron microscope that we can't afford here at Columbus Tech. I just showed you pictures of bigger universities who have. I'm not even sure Clemson and Tech has it. I don't know. I don't know where you find the big electron scopes. What universities can afford that kind of money? I don't know.
Didn't you say?
I know Berkeley has one though.
University of Florida.
Florida probably has one.
Yeah.
I think Georgia may have one. Auburn. I don't know. Because Auburn, I think Auburn's bigger than Alabama, I believe. I think Auburn has a bigger student body than Alabama does. I always thought Auburn was literature. It ain't little. But I've been to both campuses of Auburn and Alabama. And I got to admit, Alabama is a beautiful campus. That thing is gorgeous. I went to Auburn, they were building dorms and a lot of concrete trucks and cement dust in the air, and it was ugly. But now I'm not going back to Auburn since the building. It might be pretty too, but I know I went to Alabama to take some classes. It was a really pretty, it was like a big park. I never knew that. But compared to Columbus State, you know, it's nice, but Alabama it's just gorgeous. I would not mind going again just to see all the stuff on the campus. And maybe Auburn now is like that too, when they're building over there. But when you start building stuff, you make the biggest mess. But that's part of growing, I guess.
Right there you have solution.
Yeah, I made the mistake.
What are you looking at?
Blank, where it says
How many lines down?
solution's supposed to be red.
Yeah.
Oh, dissolved in, you mean the broken up solution word?
Dissolved in solution. You
Oh,
got it as a
so
blank
this
one.
word here
Yes.
should be red, okay. Like that?
Yeah.
Solution.
Well, I don't know if I need that solution to be red because when I say dissolve, I'm implying, but I'll leave it like that because it matches your thing. Enzymes, cell has enzymes, fatty acids, sugars, amino acids. Those are all the things he needs to carry out his life functions to stay alive.
What's that used to what?
Huh?
That are used to, is that supposed to be, what's that supposed to be?
Used to,
You said,
something like that. Does yours have anything at all in your sheet?
It says E.
Huh?
It
The
says
E.
Maybe I changed your sheet. What number is that?
7. That's
That's a
lesson
different
10.
lesson.
We're on lesson 9.
Oh, okay, I had wrong with that. Oh. Anything not on there? Enzymes are made of what?
Solution.
Proteins, that's protein. Fatty acids, they go with fats. Sugars go with carbohydrates. Amino acids go with proteins also. Everything's on there. I mean, everything he needs is dissolved in his cytoplasm. And you're made of gazillions of cells, and when you get sick, because your cells are malfunctioning, and you feel that as nauseated, headache, just in the depths of it. And when we give you the medications, which are chemicals, to set your cells back on their proper legs, then you recover and you feel good again. Okay? Now there are some fluids found in a nucleus. Now, are all fluids liquid? What's another fluid besides the liquid?
Yes.
Gas is a fluid. Now most of us here say, when I say fluid, you're thinking about Coke and fluid ounces and milk and water. But anything that flows and can take the shape of its container is a fluid. That includes a gas like oxygen. Nucleoplasm has a different composition than cytoplasm. And obviously from the word nucleoplasm, you know where it's found. It has no organelles. It has no organelles in the suspension. But it does have chromatin. And it has a nucleolus. And even that nucleolus is not inside a membrane. It's just a mass of stuff there. Some cells have more than one nucleolus. They have maybe 2 nucleoli. But most times you'll see a dark nucleus, and inside it, even a darker area, which is called the nucleolus. And the darker area, the nucleolus, that's the one busy making ribosomes to go outside and live their life in the cytoplasm. Now, you know there's RNA, right? And there's DNA. A lot of folks don't understand that even when DNA makes a copy of itself, it uses RNA to have it make that copy. In my 11th lesson is going to tell you, and I've learned things in that lesson that I didn't know. I don't think we knew back when I was in school. We didn't know this stuff. I'm learning things like the Tachycema segments. I never heard that word in my life. Because I've been at biology school now for 30 years. And they march on. So, and by having taught this class, the other class along, I didn't do research in biology. But now I'm digging up. I thought I knew how DNA replicated. But now I know I wasn't even close. I was close on one side. There's 2 legs of the DNA molecule, right? There's 2 strands. But one strand does duplicate the way I thought it did. But the other strand, it does it totally different. I go, oh my God, I got to teach this. And now I understand it, and I said, well, it's got to be that way. But in this case, the RNA that you see that's in that nucleolus is being used by the DNA when it needs to make a copy of DNA. It doesn't stay there. It's just kind of like you put your finger down to tie a knot in your shoe, and then your finger leaves. But RNA holds that knot, then it ties, and RNA leaves. And when you get done, you have produced what you wanted. But the RNA kind of, here, hold this here. my wife, when she's wrapping gifts sometimes, she'll say, hold this, and I will, while she does her time. And she pulls a knot out of her hand and she ties it. Well, I'd be doing what RNA does. I'm not part of that knot, am I? But without my thing in there, she ain't gonna make that knot either. And so I've discovered that. And my problem was I got into it and I spent the next 2 or 3 hours just solving my own curiosity. And I'm not doing my lesson. So now at 3 o'clock in the morning, I've learned a lot of stuff, but I ain't done nothing from PowerPoint. That's my problem. I get up on a tangent. And I'm not wasting my time because when I'm teaching, I can pull on that stuff. But I got to have things to show y'all that you can look and read. If you're like me, I got to see it to comprehend it. You can't just tell me something. Like Sunday school, they just talk to you, right? A lot of times I don't remember what verse they talked about. But they use the board and write things on there. I can't remember that stuff. Don't know why, but I do. Oh, it is not always. I'm referring to the nucleolus. When that cell divides to make 2 of itself, that nuclear membrane has got to go away. It's got to go away because I've got to take those chromosomes and make another set of them so that one set goes to one new cell and the other set goes to another new cell. Well, if the nucleus never dissolves, I can move the nucleus here, but I can't separate it. So I must remove that nuclear membrane. Then I can take the spindle, and I can pull half the chromosomes this way, and half of them that way, and cell divides. When I get done, I got 2 brand new cells, which look just like the cells that divided to make them. And that's all part of this nucleoplasm job. The only time you see a nucleus, actually, is when that cell is not divided. When he divides, that nucleus membrane dissolves, and the nuclear stuff gets to float out there among the totplasm like a prokaryote. So we know that for this thing to happen, it must revisit the years it was a prokaryote. You're eukaryote fine until you want to divide. Then you must revert back to a prokaryote and divide, and then turn back to eukaryote when you've done dividing. Because until you remove that membrane from the nucleus, you can't make half the nucleus go this way and half go that way. So there's a saying that I know is ontogeny recapitulates phylogeny.
Repeat that?
Yes.
Okay. I heard this, and when I first heard it, like y'all going, what? Ontogeny recapitulates phylogeny. And what that only means is that even us, when we are first conceived and we're real little, we have gills. We have gills. We have a tail. And as we develop, our gills go away. Now, according to anything, where did life come from?
The water.
The water. But it makes sense that you would need gills to be a water creature. Well, if our development, ontogeny, summarizes the rise of the animal kingdom. Animals came from where, we think? The water. And we had gills. So I am not surprised knowing that in the early stages of human beings, we have gills to, I reckon, honor the fact that life came from the oceans. That's phylogeny. Because if you look at phylogeny, the simplest animal of all the animals is a sponge. You know his animal? He's not a plant. He grows like a plant. He anchors in the bottom. He grows up. And that sucker's that's an animal. He produces little things that go away, swim off. They're not jellyfish. And they land on like a sunken ship, or they land on a rock or something. And then they take root like a little plant, they grow up. And the sponge, the way he actually lives, he takes in water through his sides. And then the water goes out the top. As the water comes in, he has things called amoebocytes that crawl around, and they take from the water food, and the sponge grows. And if one piece were to fall off, it will grow into, it's the simplest, there's no arms and legs, no bones. And sponges are famous, they do not grow where there's pollution. So if you were going in some bay like Apalachicola Bay and find no sponges growing, then you go, oh, that shows pollution in the water. When you find sponges everywhere, that shows the water is pristine and clear. Prince William Sound up there had the Valdez and the sponges just not coming back. And what's that tell us?
It's clear.
It's getting clear. They ain't gonna live. They are filter feeders. And if they suck in that bad chemical, they die, buddy. And for long, you won't find a single sponge. And that tells you that some factory somewhere is messing this water up. When you see a bunch of sponges, then you know things are still good. But in this thing up here about this, even a sponge, he has to reproduce somehow. But a sponge, the real sponges, you can make them wet, they'll get soft. They're good for scrubbing because they got little spicules inside them, which are they're like little sand spurs. They're good for scrubbing dishes. Of course, now it'll cost you more to buy the real sponge than to buy one made in a factory. Because to buy a real sponge, you got to pay the fisherman, you got to buy the gas, you got to buy the research. Of course, there's nothing quite like that. You can, if you have a marine aquarium in your house, you can buy a real sponge and grow it in your aquarium. The real deal. And the marine aquarium do smoke. Now, we have an aquarium upstairs. I got Goldie and Red in my aquarium upstairs. That's fresh water. Can't do much with fresh water. But man, you can get that marine water. I don't mean salt water. I mean marine. And you can put sea anemones in there. You can make a little ocean in your estimate. And at home, I used to have one. Then when I moved, I didn't set it back up. I had a big old aquarium. I had lionfish in it, and I had these real beautiful fish in the Bahamas. They're real bright and yellow. And my lionfish kill one of those fish. You can't rub a lionfish in your hand. You know why, don't you? He'll stick you and he'll mess your hand up. So when I get him out of there, I have a big old net. I use my wife's spoons, and I would kind of hurt him, and he would hit that spoon. I go, my God, this sucker. So I took him to there used to be a pet shop down by Hardaway. I said, you want this thing? I'm gonna put him in a bag. He tucked the bag. So I have him in a piece of Tupperware. And I said, you want this thing? He is dangerous. I can't put my hand in there and move around my little piece of the furniture because he'll sting me. He gave me 3 other fish for that one lionfish. And he may rip me off. So I was going to give it to him, frankly. So I'm ahead. But that marine aquarium, it took about a month for it to, you set it up, you let it run. And you check the water for pH level. And when you finally get it, after a month, you start adding your fish to it. Of course, now if you put big fish in there, you can't use many fish. So do you want either a lot of small fish or you want a few big ones? And of course, the bigger the aquarium, the bigger you do. Well, that marine aquarium, when it started recycling, it maintained itself. But a lot of folks are scared of them because the fish are they cost a lot of money. But the fish, except for my lionfish, it would kill that pretty yellowfish. It cost me about $30. That yellowfish did. And lionfish, he got bigger and bigger, and I got what I was scared of because I didn't know what he was going to do. He was aggressive.
It's like owning a lion.
He was aggressive. And they're cool looking fish. But I got scared, so I got rid of them. I didn't want some kid coming in there
Sticking his hand in the tank.
and wanting to play and want to pet the fish. of all things, yes.
For something to be characterized as an animal, it doesn't have to have a brain.
Animals, animals first off must be heterotrophs. What's it mean to be heterotroph?
They live off of organic compounds.
Well, get simpler than that. Talk to kindergarten people. A plant is an autotroph. A plant is an autotroph. I am a heterotroph. So what's that mean right there? What can plants do I can't do?
Photosynthesis.
They make their own food.
Yeah.
They can make. First off, animals cannot make their own food.
Oh, I see.
They must eat others. Okay. Now, if you make your own food, what special type of pigment do you have in your body? Chlorophyll. Am I green? No. Animals are capable of moving themselves from place to place. Can that pine tree get off and walk to my house?
So sponges can like.
Can that pine tree leave the area when the area gets polluted?
No.
Animals can. When there's a fire around, animals can flee. Trees get burned up. So motion is another trait for animals that they don't have. They both have cells, so you can't say that's a divider.
Oh, plants have a cell
So,
wall.
We don't have
I guess
cell
my
wall. We
question
don't have
should
cell
have
wall.
been, what about the sponges that characterizes to be an animal?
They are heterotrophs.
That's it.
That basically, they cannot make their own food. If they were green, then we would have to call them plants.
So the fact that they can't make their own food makes it an
That's
animal.
basic difference. Yes.
It's all right.
Now, are there some plants that eat?
Like,
Yeah,
Venus
the
flytrap?
Venus
The
flytrap.
Venus flytrap. But does he have to eat?
No,
He doesn't have to.
he just
I mean,
enjoys it.
the Venus flytrap is the kind of, he grows in soil, which typically doesn't have much nitrogen. So what he has developed, those little things are leaves. And they look like they have little eyelashes, they won't hurt you. And if you have a Venus flytrap, take a little kernel of meat and drop in there, it'll close up. It don't go snap, it just closes up.
It's not as cool.
Now what happens, what he's doing, he needs the nitrogen that's in that fly. That's what that's what he wants, to supplement what his roots are not getting. And they will dissolve the fly, and what he's doing is taking those proteins in the fly and breaking down the amino acids. And the only difference in amino acid and carbohydrate is that the amino has nitrogen,
The
the
bud
carb
does
does
not.
not. Now,
Since forever to
if
die.
that plant never saw a fly, he won't die. But if you have control, which never sees flies, and experiment you give flies to, who's going to be bigger?
One that doesn't-
The one you give flies to.
So, how does that digestion process work out? Like, where does the waste
How what?
go?
It takes them forever.
Like, where the waste
It does take a long time, but they produce enzymes like we do. You should know the name of it. It's not called pod. That little capturing device, that leaf modification,
Mhm.
it produces enzymes. But actually, and like she said, it takes a long time.
All right.
And you'll find leaves, and you'll find these little, they remind, I think a little bitty green taco shell.
Yeah.
Do you think that?
Yeah.
Little green taco shells that can actually open and close. And they have hairs going this way. They start to close so slow that the old fly don't know he's being trapped. And once the hairs do this number here, he can't get out. And then it closes on up. And then it stays closed. And he's not going, he's not, no. He just stays like that. And when he opens, it's gone. And I know Walmart used to sell Venus flytraps.
Home Depot does.
Huh?
Home Depot.
They still have some now, you know. They're seasonal. They're seasonal. And you can buy them and they won't hurt you or harm anything around you. But I always thought, well, if my house had flies, I'm not going to need a few fly traps around there. Because now there's one called a pitcher plant. It's also cool. Pitcher plants are endangered. They're in Florida. They catch you pulling one, you go to jail. It's that simple. But a pitcher plant, it looks like, you know the big old horns that blow at ball games, the big old plastic horn, make a loud noise. Well, a pitcher plant has that type of thing. Then on top, it lays over like this. There is sucrose inside there, and flies like sucrose.
So they get
And at the bottom, there's a lot of sucrose, and the hairs all point down. when the fly lands, what's he going to naturally do?
Go for the super.
Well, can he get back up? For the hair doing that?
No.
And all you do is work deeper, and then that pitcher plant will also dissolve that fly. And the pitcher plants, I've gone through Florida and seen them out there, and I tell my wife, there's a pitcher plant. And I guess you could go get it if nobody saw you do it.
Go stick your hand.
But I don't think they live in Columbus, frankly. I think they freeze and die in the environment. Had to be in a greenhouse. And I thought if I had a greenhouse and there was a pitcher plant and a game warden saw it and he knew what it was, I might be in trouble. So I don't want that risk.
In New Mexico, they were selling pitcher plants at Home Depot.
Maybe that's a species that's not endangered.
Oh.
Now I know that when I went to study things because of my credentials, I could have one as a specimen. But I got to prove that I'm an instructor and that this is for a lab or something like that. And my daddy went and did it. I know folks do it though. But they're not very pretty. They're just oddballs in the way they feed. I don't. What do you want a pitcher plant for? Except for the fact that watch what it does to a fly. Okay. But there's a lot. You know, what the question you asked about when we first began biology back in the Dark Ages, there were just two things: plants and animals. That was it. Then we decided, well, look at the fungus, look at the mushroom. It has traits of both. So we said, well, we're going to make a third area and call it fungi. And then we decided, well, we're going to look at another area, and right now we have 5 kingdoms of living things. One which is animals. All right, and we're going to get there later on when we start discussing the phylogeny. All right. Now, it's not just like Jell-O now that you can put fruit in. There's a lot more going on inside the cytosol than just a place to house organelles. There are enzymes inside it floating around to break up large particles that you take in. And then those particles that you've digested can be used by the organelles. And this PowerPoint will discuss any organelles, but the next one does. It tears them all apart with a lot of good pictures. All right. Glucose does enter a cell. It is in the cytoplasm. But the mitochondria can't use it yet. Glucose is just too big a molecule to get into a mitochondria. Now, what I did not say on here is that actually, I'm talking about a thing called glycolysis. This is glycolysis up here, without me actually writing the words on the screen. Let me tell you what's going on. Let me ask you this before I start. Do you remember how many carbons are in a glucose molecule? How many are there? There's 6. There's 6 carbons. And they have 6 oxygens, and they have 12 hydrogens. But the main thing is 6 carbons. Those carbons are big atoms. And the 6 hooked together, it won't go in, it's just too big to go into the mitochondria. So what the body does, we're going to cut you in half. I want 3, I want 3. Now these 3 can go in, and those 3 can go in. Now, when you take that glucose and you split it in half, those 3 carbon compounds, they're called pyruvate or pyruvic acid. Now, a 3 carbon compound can go into a mitochondria, but 6 carbon, this is too big. And what glycolysis does, it takes 2 ATPs to make that thing split. And in doing that, off comes hydrogen. right? Off comes carbon dioxide that you breathe out. But you get to make 4 ATPs. Now, if I'm going to sell brownies, it takes me $2 to make one. You buy it for $4. How much have I made?
$2.
Just $2. Right? I had $2. I buy my brownie mix, and I got no money at all. I make my brownie. I sell it to Haley. She give me $4. Now I'm $2 ahead, right? Well, that's what happens with glycolysis. It takes glycolysis 2 ATP molecules to make the glucose split. But when it splits, that bond's broken, right? And out comes all this energy that the cell can use to make the 2 ATPs it used and 2 more on top of it. So glycolysis actually, besides making that glucose smaller go in, you're getting 2 ATPs to the good. Now that you have the 2 3-carbon pyruvates, those can then go into the mitochondria, and then the second phase, the respiration takes over. That's called the Krebs cycle. But don't write it down. But that's another whole class and a half. But the point is this: why does the cytoplasm break that glucose down from 1 6-carbon molecule into 2 3-carbons? Why is it doing that? For what reason?
What's the enter the cell?
So it can go into the mitochondria. If you don't break up the glucose, it's just too big. It can't. You've got to get in the mitochondria to make all the other ATPs. Matter of fact, when you get done with 1 glucose molecule, you will have made 38 ATPs. It'll cost me $2 to make 38. That's a good deal.
Yeah.
That's a really good deal. But I can't even make my other 36 until I get them inside the mitochondria. So I first must spend 2 ATPs to split the glucose in half. And now this 3-carbon compound, which is pyruvic acid, and this one, they can now go into the mitochondria, and I can make my other ATPs.
What's the other one? You said this one is the pyruvate.
They're both pyruvates.
Okay.
They're both pyruvates. They're identical to each other. They're identical. So 1 glucose, when the enzymes hit it, it will split into 2 pyruvic acids. I just say pyruvate because that's what they call it. I mean, if I say pyruvate and I write down pyruvic acid, I'm saying the same thing. Saying the same thing. All right. But the real problem is you've got to find a way to get that glucose small enough that it can go into the mitochondria. And the solution is cut the sucker in half. And send half in, and send the other half in. It works beautifully. And in doing that, the cell must use 2 ATPs, but when it's all over, he's made 4. He's 2 to the good. I had 2 to start with, now I got 4.
Does that cover everything? Everything has to be broken down?
Absolutely. Mm-hmm. Well, first off, like the fructose that you're getting from apples, your body changes that to glucose and then does it. The lactose from milk, the body changes that to glucose and then does it. All the other sugars you have, the body can convert it into glucose, because glucose is the one you've got to have. You don't break down fructose, you break down glucose. But all the other sugars I can change into glucose. I wish my car did that in water. I could put water in my gas tank and change the gasoline. Wouldn't that be cool?
That'd be great.
Don't they have
Your
that?
body does that. Your body takes a non-fuel like fructose and changes it to glucose and sends you on your way. I mean, that's pretty cool to think about it. But the problem is, though, you don't need a lot of it. If you eat too much of it, you get fat. Because your body saves the extra
Convert it to
sugar
fat.
by changing them to fat and saving them. Hips get wider, fat starts to hang off of it. But if you can eat as much as you need, you'll stay nice and trim, you feel good, you won't feel tired, run down. That's hard to find that perfect barrier, though. That's perfect balance. It's hard to find that because the seasons change and you eat more, you eat less. Because when the seasons change, it starts getting colder. You do less.
You eat more.
I keep eating. I should eat less. But I keep on putting it away. And by the time spring comes, and I'm now 15 pounds overweight, I want to play tennis. I have a heart attack trying to play tennis.
It's all Christmas.
And teenagers, that high school seniors and high school graduates, they're used to a fast lifestyle. They get a job. Now they stand behind a register for 4 hours a day, and they keep eating what they did, and they gain weight. Don't know why they gain weight. I only had a whole pizza last night myself. Well, when you're in high school, you're cheerleading, and you're doing, and you can do that. But when you get out of high school and your pace slows down, you're eating, but it doesn't. But it should slow down. The problem is, though, eating is fun.
It is.
You gotta make me eat. Apple pie, you gotta put a gun in my head. Eat that pie. I can fuck that. Eating is fun. And if it tastes good, it's really fun. And that's the whole problem. How many folks said, now I shouldn't eat this last piece of pie, but
I will anyway.
I don't want you to have it, so I will. And then the next thing you say is, I knew I shouldn't eat that pie, now I feel miserable. What's that saying? Full of tick. You ever seen a tick full? How big that sucker is?
I'm waiting.
I do that.
Anyway, okay. Here's number 12. And this is just continuing what 11 started. So the enzymes in the cytoplasm rip that glucose into two pieces called pyruvic acid. And then those acids are sent to the mitochondria. And the mitochondria, you know, the oxygen you breathe in, that's where it goes, the mitochondria. And then the oxygen can take the pyruvic acid and break it on down more and make tons of ATP. So most of the ATP is made in the mitochondria. Only 2 are made in the cytoplasm when you first split the glucose in half. And that's only called glycolysis, is the splitting of glucose. So I discussed that 6 carbons being split to 2, 3 carbons. That is glycolysis. Glycolysis burns 2 ATPs, makes 4. So you're ahead by 2 really.
Is that like the Golgi complex?
Now the Golgi complex is where things are made or packaged. That's totally different concept. And the ATP down there, I'll tell you right now that you're gonna make, counting glycolysis, but those 2, when you get done with 1 glucose molecule, you will have made 38 ATP. That's a lot of money in a cell. Took you $2, and you're ahead by $36. That's a good business deal. All right.
I didn't get it on.
So that would be the end of that show, and we anticipated getting there. I want to start my next show so I can show you.